I'm interested in doing research that is quite applied, because I like the idea that what we do can actually be used in the real world. So we've tended to work in areas that are of interest to the pharmaceutical industry, and what we try to do is work out how to make protein molecules that are of interest to the pharmaceutical industry, either directly as therapeutics themselves, so things like insulin would be a good example of that, or protein molecules that they need in order to develop new drugs, so these would be drug targets. So I've got a long-term interest in things called membrane proteins, which are proteins that sit in your cell membrane, and your cell membrane keeps all the things in the right place at the right time, and many of the things that have to come into and out of cells are basically mediated by membrane proteins. So membrane proteins are like windows or doors into and out of the cell through the membrane, and you can imagine, because they play such an important role in things like letting nutrients in or waste products out, or the way that cells talk to each other, then they become important parts of healthy cells. And when the proteins go wrong, that's often associated with disease. So if you can understand the proteins and what they look like, you can then design drugs that will fit directly into the protein in some way, and that can be used for therapeutic purposes in particular disease states.
So how does that work exactly? What are you doing to the membrane itself? Tell me a little bit more detail about that.
Okay, so what we're trying to do is we're just interested in looking at specific proteins that are in the membrane. So for example, the pharmaceutical companies or other academic groups will have identified particular proteins in the cell that are known to have an involvement in a particular disease state. So for example, there might be some protein that's involved in diabetes, insulin resistance, that somebody's particularly interested in, or something like Parkinson's or Alzheimer's disease, for example. One of the interesting sets of proteins we're looking at the moment, in collaboration with the group at Birmingham, are involved in the way hepatitis C virus binds to cells. So these are proteins that sit in the membrane, and when the virus comes in, they act as the place where the virus can bind to the cell and then go on and do its thing. So if we can understand, for example, what these proteins that sit in the cell membrane look like and how they interact with the virus as it comes into the cell, then we should be able to perhaps block that interaction and stop the infection. But we're always interested in the membrane biochemistry and the understanding how to make these proteins in large quantities, and then we tend to collaborate with people who have specific knowledge in the biology. So for example, we collaborate with Jane McKeating at Birmingham University on the hepatitis C virus stuff.
I see. And are you working closely with industry in the area at all?
Yes. So most of the students that I have are sponsored by industry. So we have CASE awards, for example, and we've had a number of industry funded projects in the lab.
Okay, brilliant. And yeast, what's the yeast connection?
So the yeast is the method to actually get the proteins in large quantities. So what we want to do is to be able to get these interesting proteins, like the components of the hepatitis C virus receptor, in large enough quantities, which for us large quantities means milligrams, which is not something you can see on a spoon, but for us gives us the ability to do the analytical techniques that we need. So we need these proteins in large enough quantities that we can then go on and do our experiments. If you just went into human cells to get the proteins that you were interested in, they're present in such tiny amounts you wouldn't actually be able to get anything useful out. So what we need to be able to do is somehow amplify the amount of protein that's there naturally. So we do that by taking out the gene from a human and putting it into a yeast. And yeast are really handy because they're like us, they're eukaryotic. And they have many of the properties that human cells have, but we can grow them to very high density so we can make lots of protein. Well, people have used simple organisms like bacteria and yeast to make proteins for quite a long time now. But what we've done that I guess is a little bit different is we've tried to understand how the yeast cells themselves respond when you put unexpected proteins into them, so proteins that wouldn't normally be there. And what we try to look at is what's happening to the yeast when they're making these proteins that you want them to make, and how to make the yeast do it better, because often there'll be blockages. You're asking them to do something quite stressful, so not only are they being yeast, they're also being a factory for you, so that they're actually producing the protein that you want. And if you can actually help them to feel less stressed or cope in some way with making the protein for you, then you can make them better factories and you can get them to make the protein that you need. As you get older, all sorts of things go wrong. And so there are going to be lots of diseases that become more prevalent as we age. So therefore being able to understand what's gone wrong, identifying what the targets might be that we can intervene with new drugs is going to be something very relevant. And I think as we're all living older and there are going to be more older people as time goes by, I think this is going to be very important, I think.
And how do you see the science developing over the next few years? I mean, obviously it's developing at quite a clip already. How do you see that going over the next 10, 15, 20 years? What do you think the possibilities will be in your line of work, perhaps?
Well, I hope things will be more predictable and reproducible. So I think, especially working with membrane proteins, is being incredibly challenging because they're difficult proteins to work with anyway, because they reside in membranes which are quite oily, but they also have parts that stick out into the cytoplasm in the cell, which is more water-like. So they have very complicated properties. So actually being able to make them and handle them is a huge problem. But I think just by being able to make them in large quantities and learn how they behave in different circumstances, we're going to get a much better understanding of what they do. If we can do that and we can gain new information about them, then we should be able to use them as drug target design templates, maybe was what I was going to try and say, something like that. So I would hope that this sort of work is going to just make some of the stuff that's done at the moment, which is done in a more case-by-case specific way, is going to become more routine. And if that becomes more routine, then you open up new areas of research.